Hello and welcome. Today, we're diving into the fascinating world of archaeogenetics, a field that's like a time machine at the molecular level. Imagine reading the history book written in the DNA of beings who lived thousands, even tens of thousands of years ago. That's the power of studying ancient DNA. So what exactly is archaeogenetics? Think of it as a fusion of archaeology and genetics. While traditional archaeologists study artifacts and ruins, archaeogeneticists look directly at the genetic code preserved in ancient remains. It adds a vital biological layer to our understanding of the past. The key ingredient is ancient DNA, or aDNA. Scientists extract this genetic material from sources like bones and teeth. The inner ear bone and tooth cementum are particularly good. Sometimes, though rarely, hair or preserved skin might yield DNA. Even fossilized feces, known as coprolites, and environmental DNA from soil or ice cores can hold genetic clues about past ecosystems. But working with aDNA isn't easy. DNA breaks down over time into tiny fragments. Plus, it's incredibly easy to contaminate with modern DNA, from the people excavating the site or working in the lab. Early research often struggled with these problems. Thankfully, technology came to the rescue. Techniques like the polymerase chain reaction, or PCR, and especially next-generation sequencing (NGS), have been game changers. Combined with ultra-clean lab environments and smart computer analysis, scientists can now amplify and read even minuscule amounts of damaged ancient DNA, filtering out the modern contamination. This has led to some truly revolutionary discoveries. Archaeogenetics has completely reshaped our understanding of human prehistory. We've been able to track migrations like the peopling of the Americas, the spread of agriculture into Europe, and the movements of steppe peoples linked to Indo-European languages. We've sequenced the genomes of our extinct relatives, the Neanderthals and Denisovans, confirming that early modern humans interbred with them. Many people today, outside Africa, carry traces of this ancient genetic mixing. The field has also identified the bacteria behind historical plagues, like the Black Death, letting us study how diseases evolved. We can even see the genetic changes that occurred as humans domesticated plants, like maize, and animals like dogs and horses. And by analyzing environmental DNA from sediments, we can reconstruct entire ancient ecosystems. Of course, challenges remain. DNA preservation depends heavily on the environment. Cold, dry conditions are best, which means we have more data from certain regions and time periods than others. There are also important ethical considerations when sampling human remains, especially respecting indigenous communities. But the future looks incredibly bright. Technology keeps improving, allowing us to retrieve DNA from older and more degraded samples. By combining ancient DNA data with findings from archaeology, linguistics, and climate science, we're building ever more detailed pictures of the past. In conclusion, archaeogenetics gives us a powerful molecular window onto history. Reading the genetic stories preserved across millennia provides a direct line to understanding our ancestors, their journeys, their evolution, and their interactions with a changing world. It's a field constantly revealing secrets hidden within the code of life itself. Thanks for listening.